So Bismillahirrahmanirrahim. In this video we are going to cover the histology of iris. And if we look at this diagram, we can clearly see that this green, the very dark green layer, it is actually the vascular layer, and we know that vascular layer has a posterior portion which is known as the choroid, a middle portion from the ora serrata till the level of this ciliary processes, the ciliary body, and from there we have another process radiating outwards which is known as the iris. Now the iris has an aperture in between it, just in front of the lens, and that aperture is known as the pupil. Now this iris is going to divide our eye into an anterior and posterior chamber. So this one anterior to the iris is the anterior chamber, and this posterior to the iris is the posterior chamber. Then the process of the iris towards this aperture or opening the pupil is known as the pupillary process, and the part of the iris towards the ciliary body is known as the ciliary process. Now in this video we come to know that iris is the most anterior part of the uveal or the vascular layer of the eye, and it actually forms a contractile diaphragm, and it divides the aqueous humor, as I told you, into an anterior and a posterior chamber, and the center of both the iris have an opening which is known as pupil, and we can increase or decrease its diameter by the activity of the iris muscles. Then I told you the periphery of the iris, which is towards the ciliary body, it is known as ciliary margin. Then it is the towards the pupil, it is known as the pupillary margin. Now if we look at this diagram, we can see the histology of iris. We have an anterior side and a posterior side. The anterior side, it is devoid of the epithelium, but the posterior side actually has its own epithelium, which is further divided into an anterior epithelium and a posterior pigment epithelium. Now this anterior limiting membrane, this one present over here, which is devoid of the epithelium, on its undersurface we have the stroma. Now the stroma, it is produced from the embryonic mesenchyme, and this epithelial posterior layer, it is produced from the neuroectoderm in the embryological life of the fetus. Now if we look at this diagram of the iris, we come to know that this is the anterior most portion, then this middlemost portion is the stroma, and this posterior portion, which is further divided into an anterior and posterior portion, it is composed of non-pigmented epithelium, which is anteriorly, and a pigmented epithelium, which is present posteriorly. Now first of all, we are going to talk about the stroma of the iris. Now the stroma of the iris is actually made up of two major muscles. One is the sphincter pupillae and other is the dilator pupillae. Along with that, we have a large number of things that are blood vessels, fibroblast. Also we have certain type of melanocytes. Melanocytes are the pigment epithelium. They are going to give a dark color to this stroma. Let's have a read. Now we have the iris stroma. It is actually composed of collagen fibers. Collagen fibers are going to provide it with strength. Then we have the fibroblast for the phagocytic activity. Then we have melanocytes to impart the color, blood vessels for the purpose of providing nutrition, nerve fibers for providing control from the brain, and certain smooth muscles and myoepithelial cells. Let us have a look. So now they also tell you that the fibroblasts, the collagen fibers, melanocytes, they condense and they form an ill-defined layer, which is the anterior layer. And then we were talking about the stroma, and we found out two major muscles in the stroma. One is the sphincter pupillae, another is the dilator pupillae. So what happens that the sphincter pupillae is actually smooth muscle, and the dilator pupillae is actually made up of myoepithelial cells. Now whenever the sphincter pupillae contracts, it decreases the lens or the pupil diameter, and whenever the dilator pupillae contracts, it increases the diameter of the pupil. Sphincter is going to contract, so it is going to decrease the pupillary diameter, and dilator is going to increase the pupillary diameter with its constriction. Now we are going to talk about the nerve supply. So sphincter pupillae muscle, it is supplied by parasympathetic nerve fibers, which are coming from the ciliary ganglion through the short ciliary nerves, whereas this dilator pupillae, it is provided by the sympathetic postganglionic nerve supply, and it is mainly coming through long ciliary nerves. As you can see, the sphincter pupillae is a short muscle, so it is supplied by short ciliary nerves, whereas the dilator pupillae is a long muscle, so it is supplied by long ciliary nerves. And sphincter pupillae, as the name suggests, it has to do the sphincter, or contraction is going to decrease the diameter, so we are going to go with parasympathetic nerve supply, whereas the dilator, it is going to increase the diameter, and we know that sympathetic system is going to increase the diameter of our pupil, and it is applied by long ciliary nerves. Also, we can read it from over here that we have sphincter pupillae and dilator pupillae. The sphincter pupillae is actually made up of smooth muscle fibers, and whenever the sphincter pupillae is going to contract, it is going to decrease the pupillary diameter, and as I told you, it has postganglionic nerve fibers which are parasympathetic, and they supply through ciliary ganglion via short ciliary nerves. And the dilator pupillae, it is actually made up of myoepithelial processes, and as I told you that the dilator pupillae, whenever it is going to contract, it is going to increase the pupillary diameter, and then we have its supply from the sympathetic postganglionic nerve fibers via the long ciliary nerves. Now the radially, the dilator muscle, it is radially ranged, whereas when we talk about the sphincter muscle, it is a ring-like arrangement. Now we will talk about the two epithelial layers of the iris. As I told you, the posterior one, it is the pigmented epithelium, and the anterior one was the non-pigmented epithelium. Now we are done with this anterior limiting membrane. As I told you, it was made from certain fibroblasts, certain melanocytes, certain collagen fibers, but they were roughly arranged, and they made a very irregular type of anterior limiting membrane. Then we talked about the stroma. Then we are now going to talk about the posterior layer, which is further divided into anterior and posterior epithelial layers. The most posterior layer, which is towards the posterior side of the aqueous humor, or towards that chamber, it is the pigmented epithelium, and this pigmented epithelium has large cells, and these large cells, they are richly supplied by the melanocytes or the melanin pigments. Then we talk about this anterior layer. It is non-pigmented cells, but it has a apical as well as a basal portion. Now the portion that is towards the these pigmented cells, that portion is known as the apical portion, and that apical portion has some amount of melanocytes because it is adherent with that posterior pigment epithelium. And the anterior side of these cells, or the basal region of these cells, the basal region has myofilaments, and these myofilaments or myoepithelial cells, they're also going to contribute to the formation of this dilator pupillae muscle. So with this, we come to the apical and basal portion. The basal portion is towards the dilator pupillae and has myofilaments, whereas the apical portion is toward the posterior pigment epithelium, and it has certain amount of melanin pigments. That was about the non-pigmented anterior layer. And now this posterior pigmented epithelium also has tight junctions, so it also constitutes the blood aqueous barrier and does not allow the aqueous humor to travel from the posterior chamber to the anterior chamber through this pathway. Rather, the only pathway for the aqueous humor to move from the posterior to the anterior chamber is through the distance between the two iris, that is the pupil. Another thing that we talked about is that there are a lot of melanocytes or melanin pigments. So what is the advantage of this? It blocks the entry of light into the eyeball except through the pupil to prevent the light from being distorted into various pathways. Nature has given us only a specific pathway to allow the light to only move through the pupil and not to move from the iris or its epithelium, because it has a large number of melanocytes. Now the color of the iris, it depends upon the number of the melanocytes that are present in the stroma of the iris. If the number of melanocytes is small, then it will be a blue appearance. If the stroma contains moderate number, then the iris is gray. Small is giving a blue color, moderate number is giving a gray color, and if it is very high, then it is going to give a brown or a dark brown color. So for small we have blue, for medium sized we have gray, and for like very dark or very richly supplied melanocytes we have brown or dark brown color. With that, we come to the ending of the histology of iris. Now in the next video we will discuss the histology of retina.